Wow, it was a long day. A lot of science, a lot of information. So I thought that instead of diving deep into science again, I talk about, again, the NIH program, particularly for our patients here, what the NIH Clinical Center is providing to you. Then I give a little bit an overview of what my group is doing in regards to Degos. And at the end, I would like to talk of some very preliminary data that we think are exciting to share with you. So, my lab works on functional genomics in rare Mendelian diseases. It's a patient-centered translational program to understand and to treat vascular diseases. So, the focus, The focus of our program is the patients. So the patients is coming into clinical protocols, we bring them into the clinical center, and when we do a lot of clinical evaluation, and some of them you saw presented by Dr. Rosing. We do a lot of cardiac evaluation, vascular evaluation. We do head-to-toe clinical scanning and investigation of the patients. Then we use samples from the patients to look for genetic changes. So most of our patients that are coming in have very, very rare diseases, and we assume that many of them have actually an underlying genetic cause for that. And we are targeting, we want to try to find out what is this genetic change. However, if you look in the genome, there are so many changes that you have, and only one or two of them are causing a specific disease. So we need to find other methods, other ways to put these genetic changes in a context of the biology of physiology. So what we do what is called patient-specific in vitro disease modeling, where we take from each patient samples, and then we do all kinds of investigation to see how this patient is, based on his clinical phenotype, is different from a control patient or from an unaffected patient. And this then gives high throughput variants that then we use to strategize the disease-causing variant that we find out with the genetic evaluation. When we have this genetic variant that causes disease, we have a mechanism, we have a starting point, where then we look further in more complex disease models, how we can interfere with that, how we can treat it. So our goal is to do patient in vivo disease modeling, we use animal models, but to come up with therapies for the patients. So we go all the way from disease discovery over disease understanding to treatment strategies. And one system that we are using is stem cells. So because we think we work on genetic diseases, or that the patients have some changes in the genome, this genome is in every, or this change is in every cell. So when we take these cells into the dish and then make stem cell out of that, and then try to use these stem cells to make cells like blood vessel cells, brain cells, skin cells, it allows us to study the patient in an individual way in a dish. So we work on many different, very, very rare diseases. So these are just 4 diseases besides Degos, where we work on vascular calcification. Today there are only 12 patients known in the whole world who have this disease. It's called ACDC. DADA2 is autoinflammatory disease. These are children who develop stroke. And actually Adam we thought maybe that he had DADA2, but he was negative for this mutation. We work on SAVI, and I will talk more about SAVI. This is a malignant form of vasculitis. Again, affects children, and it is interferonopathy. And we also work on autoimmune deficiencies. Okay, so all this research that we are doing happens here. You are all here. You are probably somewhere down here in the basement over there. But the patients are In that complex, in these two units here, are the patients, and the labs where we are working very close to the patients are just here. We have 234 beds for patients. We have a lot of other patients that do not require to stay at the clinical center. They go to hotels in the surrounding areas, but we also have lodges. We have family centers. We have children inns where we take care of our patients. The important thing is to understand that the NIH Clinical Center only treats patients that are on clinical protocols. We have about 1,800 clinical protocols that treat all different kinds of rare and unusual and poor understood diseases. So the NIH Clinical Center is a microcosmos, what is out on rare diseases within a very concentrated way here on the NIH campus. We have a lot of clinical research, so everybody who works at the NIH is somehow linked to clinical research. We have 5,000 physician scientists just working on all these different diseases. And as I said, all patients are on protocol. So if you have a stroke or myocardial infarction, that you end up in the NIH is very unlikely. So we actually cannot care for these kind of patients because everything we do must be regulated, is experiments on patients. The major emphasis of the clinical center is to study the pathology of disease and to do first in human clinical trials. And many of our patient studies are on rare diseases. There are about 20 million people in the United States who have rare diseases. So overall, everybody on rare disease together, you are not alone. There are many of you. The primary mission of our clinical center is science, and all patients are volunteer. They volunteer to participate in our research and are partners in how we do the research. We cannot work here for one day if we don't have the patients here. But however, we don't only do research on the patients. If you come in here and we identify a medical problem, medical care will given to you. And very important, all the care is free for you. Any patients who comes here at the NIH, there will be no bill for you. However, it's not entirely true because you paid for all of that where you're sitting, this whole campus, this huge enterprise of clinical research by your tax money. That is the NIH. We not only see patients from United States, we basically see patients from all over the world. We get patients from Afghanistan, we get patients from India, from Siberia, all over the world. Many of these patients are flying by their own cost into the United States, and then from there, the NIH takes over and takes care of them. Again, this is protocol-centered care. All patients are enrolled in protocols, and the protocols have very strict rules to take you into that protocol. It's not a full-service hospital. It has, for example, no emergency room. We also do not have obstetric. It is a pure research facility. We do not have routine non-emergency patients in there. So now coming to our program. As Doug already alluded, we started that in 2012. So far we have enrolled in our program 9 Degos family, and 9 Degos family, including up to 7 family members. We have 13 patients with Degos disease, 4 patients with systemic Degos disease, and these are the 4 patients that are coming up again and again in our discussions that we have here. And we have 9 patients with cutaneous Degos disease. Age range for the systemic Degos, 21 to 50, I think, and cutaneous, 1 to 73 years old. And because we do genetics and we think that Degos might be inherited diseases, we not only study the Degos patient itself, we study the whole family. This could be father, mother, all the children. It could be all going over very many, over several generations. And we do a whole battery of clinical evaluation, as Dr. Rosing already alluded. We do different imaging studies like carotid ultrasound, so we look at the carotid vessels. We do different MRI studies, from total MRI to very focused MRI of certain vascular segments. We do vascular functional studies, and we include a lot of other disciplines in it. So many of our patients, for example, get eye exams, they get rheumatology exams, they get neurological exams, hematological exams. So there's a whole battery of different experts here at the NIH. But we not only do the clinical evaluation, we also use the patients and need the patients to do research approaches. So what we use from the patients, I indicated, we use samples like skin samples and blood samples. And then we do whole genome evaluation. We do blood biomarker scans to identify biomarker that would maybe used to identify certain diseases, but can also be used to stratify treatment procedures and so on. We then, as I indicated, we use these cells to make stem cells for in vitro disease modeling, the disease in a dish, and also for in vivo disease modeling. And like using animal models, humanized mouse models, they have an immune system very similar to humans, so we can actually study different processes in there. We also have a very large animal study ranging from monkeys to sheep and dogs as well, available here at the NIH. So we do not know what is really the cause of Degos. We think it might be genetic diseases, might be inherited, might be imprinted in some of our genes. There are two main models of genetic diseases. One is called autosomal recessive, where both father and mother are carriers, and their children can have different outcome based on that. It could be that if this is the affected gene, both mother and father have it, that two of their children, is just statistically, are just carriers on that. One is completely unaffected, and one of these children is affected. We cannot predict how this happens. This is just a model of certain inheritance. And the other model is autosomal dominant model, where one of the family members has one change, and this change is sufficient to cause the disease. And in this case, you would have two affected and two unaffected children. However, if you look at Degos, this scheme does not really hold through. We see many families where both parents are completely unaffected. We see some families where father has some kind of Degos, maybe not exactly the same type of Degos as the children has. And we see sometimes that in certain generations, in certain families, some family generations are skipped. So these are our 9 Degos family that we have currently that we work with. We, in black, are families with systemic Degos, and in this lighter color, we have families with cutaneous Degos. All 9 families and additional patients, some of them were actually given to us from Dr. Stratakis and were collected a long time ago in Berlin, are included in a whole genome sequencing approach that we just very recently completed. So we send them over almost 50 DNA samples from Degos patients and their family. And we just received a couple of days ago the sequencing data, which is a huge number of data that we collected, 10 terabytes. If you try to put that on your computer, don't even try it. You need some kind of large supercomputer to manipulate these kind of data. But as I indicated, the sequence information may not be sufficient for us to really find out if there is one, and if so, which one of these changes could be disease causing. So we also run on all these patients' changes in their transcription level in different cells to see if anything shows up that can explain findings that we have in the genetic evaluation. So just I want to talk briefly about the stem cell that we are using and what we're doing with them. So we use induced pluripotent stem cells. Induced pluripotent stem cells is something similar to embryonic stem cells. However, it's not coming from a blastocyst from very early stage during human development. It is actually coming from the patient itself. We can use any samples from a patient, skin samples, blood samples. We can use urine and get cells out of the urine. We can get hair follicles to get cells out of that. So all sources that are basically that we can use to be shipped all over the world to us, we can use to make these induced pluripotent stem cell. And these stem cells have the same property as embryonic stem cell. And then we differentiate them into all different kinds of other tissue systems in order to study the genetic changes that they have. So we are, and something is also important, these stem cells that we get from the patients, and because we work with very rare patients, populations where in order to get maybe 10 patients is a major thing, we need to develop systems that are very stable, that are very reliable. However, we all know that our genome and our cells are very different from each other. So if we run 10 different patients and compare them, the results may be all over the place. So how can we better use them? And one way to do that is to genetically correct these cells that we get. So if we identify a certain mutation in one patient, what we can do, we can take these cells, we can correct only this one base pair out of the billions of base pairs that these patients have. We change this one, all the rest we leave intact, the same as all the others, and then we explore how does these cells behave differently. If they behave differently, it's caused by this one mutation. If they don't, then this variant was not a disease-causing variant. What we do, we focused on mesoderm differentiation. This is a terminology from development where all the blood cells and the blood vessel cells are mesoderm derived. So I work in the Heart, Lung, and Blood Institute. That's my daily bread and butter. We use these cells to do disease modeling, to understand the disease. We use these cells to test therapies. We use them actually as therapy. For example, if you lose a lot of your muscle cell in the heart, we can come up with new strategies to regrow cells and transplant them back in the heart. And also what we do is to use these cells because they come from an individual patient for drug screening. So we can use them to predict how an individual responds to a drug before we give it actually to the individual. So this shows you how these stem cells look like. Looks like a normal cell culture. These are maybe a couple of hundred cells, and we then change them into a cell population that is present during early development. And then from there, we can easily differentiate them into stroma cells like fibroblasts, we have a lot of fibroblasts, or vessel lumen cells like endothelial cells, vessel wall cells, these are the wall cells that make the muscle layer in the blood. We can also make all kinds of different blood cells like monocytes, macrophages, dendritic cells, NK cells. We can make fibroblasts. And these are all cells that are specific to one individual that we can use to study. We also do biomark screening, and this is a biomark screen that we did, I think, 2 years ago in 2 of our Degos patients. One of the patient, DR10, has systemic Degos, and this patient has only cutaneous Degos. You see, if you look at maybe 17 different markers in the blood, and this was before the patient who has systemic Degos was on eculizumab, you see an upregulation of certain markers here. And what you see here in red is IP10, IL-5, IL-7, IL-6. So factors that you heard before might be indeed linked to Degos disease. So IP10 is a marker gene for interferon signatures. So very recently we expanded that. Instead of screening 100 different molecules in the blood, we now screen over 1,000 different molecules. We do that by what's called a Soma Scan, where if this is the protein, you see the assay is built on the generation of many different probes that are all very specific to a certain protein. And when you measure the amount of that, gives you an indication how much of any different specific protein is in your blood. So we can measure over 1,000 different proteins and quantify them at the same time in one sample. So this shows you a very preliminary evaluation of one of these experiments, where we evaluated 3 different patients who have only skin Degos to 6 control patients. And what we found, there are more than 75 statistically differentially expressed protein found in there. And what was very obvious to find was, again, an upregulation of certain inflammatory pathway. Again, IP10, which is a signature for interferon. We also find interferon gamma, and we found IL-1 receptor. Here you see a couple more. You see, what is there? Interferon gamma, IP10, IL-17. So these are all factors that are upregulated in a certain inflammatory disease, probably driven by an activation of the interferon pathway. So in cutaneous Degos patients, in 3 Degos patients, we see a statistically significant upregulation of interferon-related and NF-kappa B related signaling molecule. So we also run on 4 systemic Degos patients, and we came across these 4 systemic Degos patients before, who are all on eculizumab and treprostinil. And what we see is something that I think we would expect. We see a downregulation of the complement system. So if you see here a blue line going down, that means this is downregulated. So C5a is downregulated, something that I think you referred to earlier. We see a downregulation of C5b, and also members of the membrane attack complex. However, what is important to see is, although we see a downregulation of the complement system, and we know that these patients progress in the disease, we also see still the upregulation of the interferon pathway, meaning eculizumab, as we discussed before, affects the complement system, downregulates the complement system, but the interferon signature is still up, which is similar what Dr. Macro identified before. So, and we talked about that before, is Degos an interferon-mediated endotheliopathy? Dr. Macro said exactly that 7 years ago, where she identified an interferon signature gene, MXA, in skin samples together with the presence of the C5b-9, a component of the membrane attack complex. So what would interferonopathy do to blood vessels? What do we know about that? So now I bring you to a disease that we understand much better as Degos, which is called SAVI. This is a sting-associated vasculopathy with onset in infants. This is one of these children who has these changes in her skin, in her fingers, on feet, on her nose, on her cheeks, in her ears. And these are just very mild version of that disease. If the disease is progressing, the children are losing their feet, they are losing their finger, they are losing their nose, they are losing their ears. It's a very devastating disease. It's a very malignant form of vasculitis. And if you look at the changes that these children have in the skin, it's actually not so different from what we see in Degos disease. However, these children also develop an interstitial lung disease, which is the clinical most relevant changes. And some of these children, I think 30 or 40% die because of the interstitial lung disease. This disease is an interferonopathy. You see an upregulation of the interferon signature gene here in the SAVI patients compared to the controls. And the disease was identified in the lab of Dr. Goldbach-Mansky, Rafaela Goldbach-Mansky, who is an investigator at NIAID. And we were working together to better understand this disease. What is the STING pathway doing? So the STING pathway is actually part of the viral defense mechanism. So when a virus goes into the cell, the viral DNA is exposed to the intracellular machinery of the cell, and we have defense mechanism against that. And one of these defense mechanisms is STING. It senses the DNA, the viral DNA, and then upregulates interferon to fight the viral infection. However, in these patients, all the cells that have these mutations think they are viral infected all the time and produce enormous amount of interferon. These high levels of interferon lead, as I indicated, to an interstitial lung disease, which is the most cause for fatal cases of SAVI. This interstitial lung disease is very particular. It's very different from other interstitial lung disease, as you see a very strong perivascular fibrosis. And some of these changes are very similar to what you actually see in patients with scleroderma. And some of the changes that we saw in the Degos patient in the pericardium have also a very similar appearing. So running through a couple of studies, what we identified is that in endothelial cells, you have the activation of the viral defense mechanism that increases interferon. If these patients have any infection in their lung that leads to additional interferon beta or alpha production, these situations, these between extrinsic and intrinsic factors, leads to a scenario where the endothelial cell actually doesn't stay an endothelial cell. It changed the phenotype to a fibroblast, to a very different cell type that then causes the fibrosis in that. Why did I tell you all of that? It's interferon-driven. Fibrosis is a main component on that. There are similarities to other interferonopathy. There are similarities to scleroderma. I think there are similarities to Degos. We know that when we in experiment block this interferon activation with a check inhibitor. Experimentally, we can block this process. In the patients, we can stop, at least what we know so far, the progression of the disease. So in summary, Degos is a devastating disease with still unknown cause and mechanism. First, treatment approaches are encouraging but not sufficient. We need to increase our effort in basic and clinical research to improve the outcome for our Degos patients. And we need a participation of Degos family, care provider, patient advocate groups in a combined effort to better understand, diagnose, and treat patients with Degos disease. Thank you. With the last slide, I'd like to thank all Degos patients and SAVI patients that contribute to that, the people in my lab, the people who do the clinical work here, many of them are here as well. Also our collaborator at NIAMS at Cowan, our collaborator Goldbach-Mansky at NIAID, the sequencing core over at USES, and all other people. It needs a village to raise the child, it needs a village to bring science forward as well. Thank you very much. Yeah.
I fully agree that the way to go is to statistical studies. The way to go is to statistical studies. I fully agree. That's your vision. Are we closer to general diagnosis or to a polygenic disease that and considering the number of your samples and your statistical power, that will be challenging falling to the polygenic disease rather than polygenic for the few genes.
So at the moment, the whole genome sequencing is very successful. It's very successful for single Mendelian recessive diseases. It's quite a challenge if you want to tackle autosomal diseases, where most of the people who have it actually also who have the genetic variant, also have the disease. However, if you look at Degos, none of that really fits. So we have Degos patients, different generations, different disease progression, disease grade, that probably an autosomal disease with a low penetrance could be a potential model. And then you need many patients, many families identify each. It could also be a recessive pattern, where you have a very rare variant combined with a very common variant. Then you see these pattern in generations that could mimic something like an autosomal dominant disease. However, what we put all our effort to not only rely on the genetic information, but also rely on the functional genetics. Find out what are the changes in the specific cell types, what are the signaling pathways activated there. Look at the single cell level, and look at one cell. Now have technology to really identify what is happening in a single cell, and you can evaluate almost 10,000 cells at one time, then combine them with high throughput biomolecular and with the clinical data, because it makes no sense to look into an eye cell and a vaginal cell when the disease is not a disease. So we need to use the clinical data to identify our cellular target, and use the cellular target to guide us in the genetic evaluation. So this is our strategy.
So actually, your samples, what type of cells are those? Who was cells or indicators? We are using the samples.
So we get, it's an indicator, we get blood cells, we use these blood cells, we change them to stem cells, and then we change the stem cell from 20 cells into smooth muscle cells. into fibroblasts, into myeloid cells, and then we put the system back together and put different components. We use fibroblasts from the normal patients together with an individual cell from the patients and try to generate a system that makes it available for us to identify changes.
Right, so these are expression, signature expression studies in your whole genome, your type of cells.
Oh, for the whole genome?
Yeah.
Yeah, we use blood cells.
Blood cells.
Of course, if it's not in blood cells, if it's a mutation that is happening later on in development, we would use this, but we don't have any mutation for that. It's endothelial cell kind of endothelial cells are very early to development. So using blood cells is going to be good.
So finally, your hypothesis is that something that happens, that is, the penetrance is very low, and happens later in life, when a 4 years old, as a mutation of some genes. Some cells, this is why it's so elusive, so difficult to catch. This is what I think.
I think about CADASIL. CADASIL is a disease, it's a rare disease. We know the mutation in CADASIL, it's a mutation in bloodstream. It's almost exclusively expressed in Soma Scan. And the patient, it's a vascular disease in the brain. And the patients become sick when they are 30, 50 years old. Children are completely normal, they don't have any phenotype. It can take decades to develop even a disease that is inherited right.
Thank you so much, and so thank you also for communicating what you do here. I may speculate, but I have a strong feeling that it's not too far from the truth. This incredible variant phenotype, this incredible variant severity of the disease, indicates clearly that it's not a genetic disease. I don't believe that this is a gene that makes all these variations in those different ages in this kind of.
One example. The other two is another disease we identified with Degos a couple of years ago. It's a deficiency in DDA2. The disease population was identified as children who get strokes. Then another group find out the same mutation causes pain, and release nervosa in older patients. Then we find out another population who develop leukemias. Then we find another population who develop Alps syndrome. Then another patient population develop immune deficiency. And all these very different kinds of diseases, leukemia, vasculitis, anemia, immune deficiencies, are caused by the same mutation in the same gene.
Etiologically, we're sure that there's an etiological reason of all these variations.
We have no idea how this can develop. This is very complex. Cell interaction can have certain effects in the bone marrow, changes there, can lead to vasculitis, some adults, can lead to stroke. So I would not exclude that different organ systems are affected, because we know that Degos disease induces vessel occlusion, induces stroke, it's the main pathognomonic. Why it affects different organ systems? We need to do it. Why does CADASIL affects most small vessels in the brain? And heart, liver, in kidney, or blood, genes expressed everywhere.
The other side is the technology you have developed via the IPS pathway. And I know it's because we do the same story for Alzheimer's. You get a very good expression of differential expression of genes as far as you differentiate the IPS to different tissues. It's very peculiar, at least in our work, that if you differentiate the cells to central nervous cells, you get the same genes that you have in the patient. If you differentiate the IPS to different tissues, you get a much less expression, differential expression indicating his difference. Because that's a powerful model that I could imagine that if you concentrate on that, then probably if it is a vascular disease, you're going to get much stronger differential expression at the cells that can be differentiated to vessels, and even maybe you make microvascular large it's not today is doable, than cells that differentiate to pseudocytes or cells that differentiate to keratine cells. And then, of course, you get more and more information of differential expression by the same very few patients by making this gain. So I would, from my feeling, maybe totally untrue, but I have this feeling that the work on the IPS pathway, having more and more patients, will be much more successful, getting a bunch of genes that can be regulated on a specific tissue than working with genetic studies.
Yeah, so we think we need to go both ways because you can never use IPS cells in the diagnostic tool. Even if you identify a change in the IPS cells, you can only have this change in the IPS cell if patients are controlled, if there's a genetic defect. So everybody will ask, show there are changes, but what is the cause? And the cause is genes in this case. And it might be something